Hello, my name is Ting-Ting Wu. I'm an associate professor in the Department of Molecular and Medical Pharmacology. There's no better time to work on vaccine, and a big part of my lab is to study vaccine in the context of a cancer associated herpes virus, more recently influenza viruses. So the major platform we had in the lab is based on lab attenuated virus. So the idea is to remove the ability of the virus to contact type-1 interferon response, which is the first line host defense against viruses. So this strategy creates a recombinant virus that no longer efficiently replicate in the host where there is a type-1 interferon response. The bonus for this strategy is that now the recombinant virus become efficient at inducing type-1 interferon, which is important for priming the adaptive immune response like a T cell and an antibody. So we perform high throughput genetic mutagenesis over the across entire full genome, and we identify multiple mutations that make the virus sensitive to interferon. So we combine those mutations to create this recombinant flu virus. Now is not only attenuate, but also very efficient at stimulating a protective immune response. So my group and Dr. Renson's group is part of this joint effort for universal flu vaccine. So research at our university apply computational approach to design a flu antigen that potentially induce broadly neutralizing antibody across different strains. So we are going to use our live flu vaccine platform to express this antigen. So one of the rotation projects available in the lab is to construct these viruses and characterize those flu recombinant virus in vitro as well as in vivo, and analyze their antibody and T cell response and help us to see whether the virus can be used for a vaccine. And so one of the things about live virus so immunogenic is because they are very efficient at alerting the host immune response or immune system. But if we can identify a molecule mimicking the ability of the virus to induce inflammatory response or immune stimulating property, we may not need a real virus for vaccine. And this is like a concept for the adjuvant. So my lab is also collaborating with Dr. Kaya's lab to explore the potential of a small molecule that targets nucleotide metabolism to enhance immune response of a vaccine, particularly in elderly population. And so the other rotation project is to investigate or determine the impact of those small molecule and vaccine not based on live virus, but other type of more safer vaccine platform, such as the mRNA or chemically inactivate whole viral particle. And those type of, so we were hoping that this small molecule can help enhance immunogenicity of the vaccine not based on live virus. Okay, so with this rotation project, you will learn the biology of a flu virus and mouse infection model, as well as immune response. And I have a relatively small lab. I like to keep this way because I like to frequently and closely interact with my graduate students. This is what I like to do. So if you are interested in any of those 2 rotation projects I just presented, please email me. I'm looking forward to speaking with you and answering a question you have for my presentation. Thank you.